sorry ass DJ.
Hello everyone. Today, we will discuss 3 topics. First, how viruses invade the cells. Second, the multiple brands of vaccines. And lastly, the 2 types of vaccines existing in our country. So, are you ready to learn more?
How viruses invade the cells. Viruses bring their DNA and RNA instruction to the cell and trick the cell into following them. The cell follow the virus direction and make all the necessary parts for the virus. Cells even use their own tools and raw materials for the virus parts. New copies of viruses can be then put together inside the cells. Best example of this is the COVID-19, which continue to exist in our society.
There are the 5 steps of how viruses invade cells. Step 1, attachment. Step 2, penetration. Step 3, uncoating and replication. Step 4, assembly. Step 5, ingress or release.
I will explain the 5 steps of how virus invade cells. Step 1 is the attachment. Influenza virus becomes attached to a target epithelium. Step 2 is penetration. The cell engulfs the virus by endocytosis. Step 3, uncoating and replication. Viral contents are released. Viral RNA enters the nucleus where it is replicated by the viral RNA polymers. Step 4 is the assembly. Viral mRNA is used to make viral proteins. And the last step is egress. New viral particles are made and released in the extracellular fluid. The cell which is not killed in the process continues to make new virus.
How to stop viruses from invading the cell? We can stop the virus in mutating by injecting antibodies, which we call the vaccine. It is a substance used to stimulate the production of antibodies and provide immunity against one or several diseases.
Why are there multiple COVID-19 vaccines? First, diversity is important in vaccine development. Producing more than one vaccines also means that manufacturing delays become less risky. With the world relying on multiple companies to produce the life-saving products, delivery delays of vaccine can be offset by the production of other vaccines. Next, cost is another reason for multiple vaccines. High-resource countries having pre-purchased millions of different vaccines directly from distributors to immunize their populations, which is not possible for some countries. Lastly, vaccines need to protect diverse group of people. Every person will respond differently to each vaccines and have different immune response. So having a variety of vaccine types fill these needs is a more concrete strategy than relying 1 or 2 vaccines alone.
2 types of vaccines. Vaccines have 2 types known as traditional and mRNA. Traditional vaccines consist of entire pathogens that have been killed or weakened so that they cannot cause disease. Such whole pathogen vaccines can elicit strong protective immune responses. Many of the vaccines in the clinical use today fall into this category.
mRNA vaccine is a type of vaccine that uses a copy of a molecule called messenger RNA to produce an immune response. The vaccine delivers molecules of antigen encoding mRNA into immune cells, which use the designed mRNA as a blueprint to build foreign protein.
So in this video, I will be in charge to introduce or explain about messenger ribonucleic acid or mRNA vaccine and traditional vaccine. So first is mRNA vaccine. mRNA vaccine are newly available into the public but have been studied for decades. The researchers have been studying and working with this mRNA vaccine for decades. Interest has grown in this vaccine because they can be developed in laboratory by using readily available materials. This means vaccine can be developed and produced in a large quantities faster than with other method for making vaccine. mRNA vaccine have been studied before for flu, Zika, rabies, and cytomegalovirus or CMV as soon as the necessary information about the virus that caused COVID-19 was available. The scientists began designing mRNA instruction for the cell to build unique spike of protein into mRNA vaccine. The future mRNA vaccine technology may allow one vaccine to provide protection against multiple diseases, thus decreasing the number of shots needed for protection against common vaccine-preventable disease. Aside from vaccines, cancer research has mRNA to trigger the immune system and to target the specific cancer cell.
How traditional vaccines work. Conventional vaccines usually contains inactivated disease-causing organisms or proteins made by pathogens or antigens, which work by making the infectious agent. They stimulate the body's immune response, so it is primed to respond more rapidly and effectively if exposed to the infectious agent in the future.
How mRNA vaccines work.
Instead, mRNA vaccines use mRNA created in a laboratory to teach our cells how to make a protein, or even just a piece of a protein, that triggered an immune response inside our bodies. That immune response, which produce antibodies, is what protects us from getting infected if the real virus enter in our bodies.
Here are the differences and similarities of vaccine. The components of mRNA vaccine is the blueprint of protein, while the components of traditional vaccine is the microbial process or inactive response. The production of mRNA vaccine is faster because mRNA molecules are easier to produce, while the production of a traditional vaccine is slower and more difficult to produce the right type of protein. However, the process of mRNA vaccine, the components are injected into the arm and serve as instruction for the body to make microbial protein. While the process of traditional vaccine, the components are made in a lab and injected to the arm to stimulate immune response. Therefore, the similarities of mRNA vaccine and traditional vaccine is the R&D, or the antigen determined for immune stimulation, and the results, it teaches the body to protect itself against a microbe.
And that's our lesson for today. If you want to learn more, please subscribe in our YouTube channel to be updated on our next learning video. See you soon. Bye-bye.